Okay, the small intestine. The small intestine, it's the longest segment of the digestive tract. It's a tubular organ. It's very long. It's about 6 meters, right? Which is extremely long compared to your height. Okay, so this tubular organ extends from literally the pylori sphincter to the entry of the large intestine. It fills most of the abdominal cavity. Now, the small intestine is folded. You remember I told you about the mesentery earlier, the two sheets that are brought together, and where you're going to have some blood vessels that run through and some nerves that also run through, right, to actually innervate the digestive tract and to nourish the digestive tract, right? So we are going to see that again. So the small intestine is actually going to receive the chyme that comes from the stomach. Remember that chyme is acidic. The chyme is going to be dumped in a part of the small intestine called the duodenum. This is also where the pancreatic juice and the bile salts are going to be released. And remember that the pancreatic juice has bicarbonate, which is a salt that is going to actually neutralize the acidity of the chyme that comes from the stomach, right? So this is also in the small intestine where not only the final digestion chemically and mechanically is going to happen, but this is where all the small building block of macromolecules are going to be released, and they're going to be absorbed as nutrients. As they are absorbed, they are going to be transported to their location. We are going to address that a little later. So there are three parts to the small intestine. We're going to have the duodenum, which is where the chyme is dumped, right? We're going to have the jejunum, which is the part that comes after. And we're going to have the ileum, which is close to the large intestine. Okay? So the duodenum, so this is where the pancreatic juice is going to be dumped. Okay? The jejunum, it's the most active part along with the duodenum in terms of digestion and absorption. The ileum is actually less active than the jejunum and the duodenum. Makes sense because as you go toward the large intestine, you are starting to see there more and more of what is going to become feces at some point, what is going to become waste at some point. So the jejunum and the ileum are going to be folded because of a structure, a peritoneum structure called the mesentery. I told you about that earlier. Okay? And in the mesentery, we're going to have bunch of blood vessels and nerves and so on. So the nerves are essential for innervation of the muscles, right? And then we are going to have actually a part of the peritoneum called the greater omentum. That greater omentum is going to actually wrap the stomach and the large intestine. Okay? So here we have the mesentery, okay? And you can see here the mesentery right here as a structure where you're going to have some blood vessels and nerves too. Now, when we zoom in, so here we are in the submucosa, and here we are in the mucosa. Now, notice that you have blood vessels right here and right here. These are venules right here, and right here we have arterioles. And here we have a capillary bed. We have a capillary bed. That capillary bed, remember, the capillaries are characterized by the fact that they are only made up of one layer of tissue, a thin, flat epithelial tissue, simple squamous epithelium. That structure is compatible with exchange, right? So the nutrients are going to leave the lumen here to be transferred or transported to the blood through the capillaries. Okay? Now, the blood capillaries are going to absorb, are going to accept anything that is nutrient that is hydrophilic. Any nutrient that is hydrophobic is going to be accepted by some capillaries, not of the blood, but capillaries of the lymphatic vessels, okay? Specifically called lacteals, right? This is one lacteal in green here, okay? The lacteals are the one that are going to absorb the fat, okay? Notice that we have a simple columnar epithelium here. And that simple columnar epithelium is going to have some goblet cells embedded. So we're going to have a basal end of the cell. We're going to have an apical end of the cell where we're going to have some foldings, right? Allowing the surface area to be increased multiple fold, thousands of fold, right? Which allows a good exchange of nutrients. So I was telling you about the basal part of epithelial cell and the apical part, apical top. So we have apical side. So this is where we have the lumen. Okay, this is where the digestive food is going to be found. And here we have a basement membrane, right? That's the basal part. Now, you have the microvilli, okay? You're going to have the nucleus. You're going to have endoplasmic reticulum, the Golgi apparatus, all of these different organelles in there, okay? Absorption is going to occur here toward in the cell, okay? So here we have a micrograph of the apical end and the inside of a epithelial cell, okay? A columnar epithelial cell in the intestine. All right. Now, what are the major digestive enzymes? the major digestive enzyme here, actually what we are doing here is to summarize what has happened from the mouth till the small intestine. So we're going to have the salivary enzymes. The salivary enzyme, we're going to have the salivary amylases that break down polysaccharides. I want you to remember that, okay? So the salivary glands are going to contain the salivary amylases, okay, which means that the carbohydrates will begin their digestion in the mouth in terms of chemical digestion, obviously, right? So here the polysaccharides are broken down into disaccharides. An example of polysaccharide is starch. Another one is glycogen. Remember, starch from vegetables, plants, glycogen from animal origin. Then we go in the stomach. In the stomach, we're going to have gastric enzymes. Among those gastric enzymes, we're going to have pepsins. Remember, pepsins are active forms of pepsinogen, and pepsinogen is activated by hydrochloric acid. Remember that, right? Okay. Now, along with pepsin that breaks down proteins, you're going to have gastric lipases. We didn't mention that earlier. We're going to have gastric lipases. The gastric lipases are actually starting the digestion of fat. However, that digestion is going to be limited because of the level of emulsification, right? We don't have as much emulsification of fat in the gastric area than we do in the small intestine, because at least in the small intestine we have the bile salt. And then we have the pancreatic enzymes. The pancreatic enzymes are going to include the pancreatic amylases, which again are going to target polysaccharide, specifically starch and glycogen, and break them into disaccharides. Then we're going to have the pancreatic lipases. They're going to break down triglyceride into fatty acid and glycerol. Trypsin and chymotrypsin are going to both break down proteins, okay? And they break down these proteins into peptides, which are small chunks of amino acids connected together through peptide bonds, right? And then we're going to have carboxypeptidases that are going to break down peptides into amino acids, which are the building blocks, right? And then the nucleases are going to break down nucleic acid into nucleotide. Remember, the nucleotide are the building blocks of nucleic acids. Amino acids are the building blocks of proteins. Glucose is the building block of starch and glycogen, while fatty acids and glycerol are the building blocks of triglyceride. So you have the whole thing here, right? And then you have the intestinal enzymes. Among the intestinal enzyme, we're going to have the enterokinase. The enterokinase are going to be essential for converting trypsinogen into trypsin. Remember, trypsinogen is an active form of trypsin, right? Enterokinase, it's an enzyme that is going to actually activate trypsinogen to turn it into trypsin. Then we have the peptidases. The peptidases are going to break down peptides into amino acids, the building blocks. And then we're going to have some lipases in the intestine that again are going to break down the triglycerides into fatty acids and glycerol. And now we have some enzymes we had not talked about until now. Sucrase is going to break down sucrose, which is a disaccharide, right? Maltase is going to break down maltose, which is another disaccharide. Lactase is going to break down lactose, which is another disaccharide. So we have sucrase breaks down like table sugar, right? Which is sucrose, and sucrose, it's glucose and fructose connected together through a covalent bond. Please remember that. Maltose, it's two molecules of glucose attached together through a covalent bond. While lactose, it's glucose and a galactose molecule attached together through a covalent bond, right? So they are very specific. The enzymes are very specific as to what kind of molecule they are going to break down. Sucrase can only break down sucrose, doesn't know how to break down maltose. Okay? Likewise, maltase knows how to break down maltose, doesn't know how to break down lactose. By the same token, lactase knows how to break down lactose, but doesn't know how to break down maltose or sucrose. Okay? Very important to remember all this. Now, the bottom line here, and what I want you to remember, it's that when you eat food, when you ingest that food, which is a chunk of steak or burger, burger or chicken or even salmon, right? This is going to be broken down into the simplest building blocks that make up the macromolecules of life. Let me repeat that. When you do digestion in your system, at the end of the day, when everything is said and done and digestion is complete, you only have, at the end of the day, amino acids from proteins, you have nucleotides from nucleic acids, you have simple sugars, including glucose, from polysaccharides, and you are going to have fatty acids and glycerol from triglyceride and more complex lipids. All right? Very important to remember all of that. And that segment is important because now you understand that your system can never ever absorb whole proteins. That is not possible. Okay? Your system can never absorb whole proteins. Your system cannot absorb polysaccharides. Okay? They have to be broken down into small pieces. Okay? So, in the small intestine, absorption is going to be optimized by a large surface area that is made possible because of the villi that fold into microvilli. That is very important to remember. Okay? So the villi increase the surface area for absorption. Very important. Okay? So the small intestine is where most of the absorption is happening. Okay? This is where most of the absorption is happening. For instance, the sugars, a sugar such as maltose, which is a disaccharide, glucose and glucose, broken down here by maltase, right? Now we have one molecule of glucose, another molecule of glucose. This is what the small intestine can absorb and transfer to the blood. All right. Protein digestion. Here we have a dipeptide. Okay? That dipeptide has to be broken down. Okay? It has to be broken down right here into one amino acid and another amino acid. So now you have an amino acid and an amino acid. All right? And that's the form that is being absorbed by the body. How about the fat? Here we have a triglyceride. Okay? We have glycerol and fatty acid 1, fatty acid 2, fatty acid 3. You need lipases that break down, releases a molecule of glycerol, a fatty acid here, another one here, another one here. That's what is absorbed by the small intestine. Okay? Please remember that. Now, the fatty acids are tricky in terms of how they get absorbed. Well, the fatty acids typically are going to be absorbed through the apical area. Okay? Through the apical area, these fatty acids are going to end up inside of the cell, but inside of the cell, because of the water-based environment, these fatty acids cannot just stay like that. Okay? They will have to be combined with some proteins. These proteins are going to form structures along with the fatty acids. This structure is called kilomicrons. These kilomicrons are nothing but lipoproteins. These lipoproteins are these lipoproteins are water soluble. Okay, they are water soluble. Okay, they are the one that can leave the cell through the basal area of the epithelial cell. From that basal area, the kilomicron are going to migrate in the lymphatic vessels and specifically the lacteal, right? And from there, the kilomicron are going to migrate toward the blood and eventually end up in the blood. So, the absorption in the intestine is going to happen as follows. The nutrient monosaccharide are going to be transported in the blood by facilitated diffusion and by active transport. Okay? By facilitated diffusion and by active transport. This is how sugar such as glucose is transported in the blood capillaries. Amino acids. Amino acids are transported in the blood capillaries via active transport. Okay? No facilitated diffusion for amino acids. How about fatty acids? Fatty acids are transported via facilitated diffusion of glycerol and diffusion of fatty acids in the cell, okay? And eventually they're going to combine with proteins to form structures that we call kilomicrons, and these kilomicrons are now going to go in the lacteals, right? Now, some fatty acids that are very small can actually go through blood capillaries. Then we're going to have electrolytes, okay, such as iron, sodium, potassium, all these ions, right, are going to come in by different ways. Some are going to be able to diffuse, some substances are going to be able to diffuse, some have to be carried, actually most, I should say, have to be carried via active transport. Okay? And then water is transported by osmosis, okay, in the blood capillaries. And the electrolytes are also transported in the blood capillaries. So really, only lipids are carried in lacteals. Now, the movement that occur in the small intestine, we're going to have peristalsis and segmentation. Peristalsis, wave-like motion, okay? While segmentation, it's ring-like, all right? What else do I need to mention here? So that's basically, what you need to remember here. One thing that is maybe, worth mentioning is that the parasympathetic system in the nervous system is the one that controls the movement. Remember that the movement in the digestive tract past the mouth is involuntary, right? So, therefore, it has to be the parasympathetic system that is going to send the impulses here for this. Okay, so the parasympathetic system, I mean, one, two, three, para parasympathetic impulses are going to stimulate the mixing and peristaltic movement, while the sympathetic system is going to actually inhibit. In other words, when you are actively digesting food, the, the parasympathetic impulse is the one that is going to come into play. And when you need to stop digesting food, the sympathetic system is the one that is going to actually send impulses to stop the peristaltic movement. All right, so now the ileocecal sphincter. The ileocecal sphincter is a sphincter that is located between the ileum and the cecum, which is the first segment at the entry of the large intestine, right? That ileocecal sphincter controls when whatever is remaining of the food that was digested is going to be there so that it can move to the large intestine and start being sent toward the exit. When I say exit, I mean elimination. Now we are in the end intestine. So we have the ileum here, and we have the ileocecal sphincter here that regulates the movement, right? So the chyme that was here going to be dumped right in here. And in here, actually, we are going to have different movement. We have the chyme that is going to go this way, this way, this way, this way, and finally here and here. Now, specifically, you are going to have a number of segments here of the large intestine. Now, we have the cecum right here. Then we're going to have the ascending colon, the transverse colon, the descending colon. Here we're going to have the sigmoid colon right here. This is the rectum. This is the rectum right here, where the feces is stored, and here the anus, where the feces is eliminated. So we have here a structure of the bottom of the large intestine. With here we do have a sphincter here, actually, that controls when to release the feces or not when to release the feces. And this one is controlled voluntarily, okay? This anal sphincter is controlled voluntarily. The rectum, it's where the waste, the feces, is going to be stored. Now, the large intestine, just like the small intestine, is going to have some goblet cells, okay, that are going to release the mucus. So, in the large intestine, at this point, there is almost no digestion happening, okay? All the digestion has been done. Most of the absorption has been done. But we're going to have some absorption that is going to be done here, and particularly the absorption of vitamin K, vitamin B12, the thiamine. Okay. Now, in the large intestine, this is where you're going to have a lot of the normal flora. Okay, that is going to be there to help digest some of the food that the small intestine did not digest. Now, the large intestine is feeding on, not the large intestine, I'm sorry. The normal flora in the large intestine is feeding off of the waste that your system sends in the large intestine, right? The byproduct, some of them are actually absorbed in the large intestine. For instance, vitamin K. Now, E. coli, which is a bacterium, E. coli, it's actually a bacterium that produces vitamin K in the large intestine. Okay? All right. So now we're going to have the feces that is going to be discarded. Now the feces, it's bunch of water, a lot of ions, a lot of electrolytes, right? Mucus, bunch of bacteria, bile pigments. The bile pigments are the reason why the feces has the color they have, right? And you are going to have a number of chemicals that cause the funny smell of the feces, including phenol, hydrogen sulfide, indole, skatol, ammonia, and all these chemicals byproduct of metabolism performed by bacteria in the large intestine. Now, what are some issues of the large intestine? Some issues of the large intestine, health issues, it's for instance, diverticulosis. Diverticulosis, this is basically when you have a weakening of the wall in the large intestine. This typically actually leads to protrusions, okay? Protrusions, and these protrusions are going to cause pouches, okay, that are going to become inflamed and eventually can become infected, which is actually not a good news. Then we have the inflammatory bowel disease. This inflammatory bowel disease is actually not one disease, but it's a group of disorders. This includes, for instance, the ulcerative colitis, right, that typically affects the mucosa and the submucosa of the large intestine. This can cause diarrhea, cramps, bloody diarrhea and cramps. You have the Crohn's disease. That is relatively nasty, not relatively, it is nasty. Okay, the Crohn's disease that affects relatively all layers, not just the mucosa and the submucosa, but it can also affect the muscularis. Okay, and this can affect both the large intestine and the small intestine. Okay, cause diarrhea and pain. And then we have the colorectal cancer. The colorectal cancer, this is basically the cancer of the rectum, okay, and the colon. So the colorectal cancer is one of the most common cancers in the United States. There is a screening that can be developed when basically there is a history of colorectal cancer in the family, right? It's a good idea to actually do a colonoscopy, right? To actually see along the colon if there is any mass that that can hide maybe some cancer there. Okay, so this is what we had for this chapter, and we are going to stop here. Thank you for your attention. If you have any question, please let me know. Again, thank you for your attention. Bye now.